Okay, so here we are at the end of our segment on mitosis and meiosis. We just finished up meiosis 2. So in this last short video, I just want to recap the main points where we see genetic variation. I'm going to tell you about two places you've seen already and then introduce a third. And the last thing we'll look at is what can happen when you get errors in meiosis. So let's go back to the beginning and think about genetic variation. The first place you get genetic variation in meiosis and sexual reproduction is when you do homologous recombination, also known as crossing over. So as what we have here is, on the left, a pair of homologous chromosomes, and each chromosome has a pair of sister chromatids, like we talked about when we talked about meiosis. And so during early meiosis 1, the two homologous chromosomes will form what's called a synaptomemal complex. This is where the two homologs come together. When they're completely associated, they form a structure called a bivalent or bivalent, and then we're going to see a reciprocal exchange of information. So that's when information on one of the sister chromatids will be swapped with information on the sister chromatid in the other homologous chromosome. So if this is the chromosome from mom, and this is the chromosome from dad, and these are the sisters, so what we're going to see is a reciprocal exchange of information, process called crossing over, and the structure where that forms is shown here on the right hand side, called a chiasma. And so it's a reciprocal exchange, it's a mutual exchange of information where information from the paternal or maternal chromosome is exchanged with the other chromosome in the homologous pair. This is like shuffling a deck of cards, basically. It's short, it mixes up and sorts the genes that would be on the pieces of DNA that are getting exchanged. So that's the first place we saw genetic variation. And then the second place we saw genetic variation is in metaphase 1 of meiosis 1, obviously. And remember, there is what's happening is that the homologous pairs of chromosomes, one from mom and one from dad in their replicated form, line up on the metaphase plate. And then we can have either mom on the left or dad on the right, so homologous chromosomes from mom on the right and homologous chromosomes from dad on the left, and homologous chromosomes from dad on the right. They can either line up like this, or they could be the other way around, so they could look like this. And so depending on what pattern of pairing up we get during metaphase 1, we can produce cells with different combinations of maternal or paternal chromosomes. So that's the second place, is independent assortment. That's going to be important when we come back and we look at some of Mendel's laws. So make sure you understand that now, because it's going to come back in a little bit. Okay, the last place that we see genetic variation is in the act of sexual reproduction. So sexual reproduction, all things being equal, is a random process. There's no way of knowing which sperm produced by meiosis and which egg produced by meiosis are going to come together. And so that randomness gives you a huge amount of genetic variability. So when we combine the crossing over, the independent assortment, and the randomness of sexual reproduction, we have a huge amount of genetic diversity that we see in populations that reproduce sexually, producing their gametes by meiosis. So the last thing we're going to take a look at here really quickly is what can go wrong when you see errors in meiosis. And so this kind of error in meiosis can lead to genetic defects, conditions such as Down syndrome, for example. So let's take a look at this pretty quickly, and we're going to look specifically in an event called non-disjunction in meiosis 1. So disjunction is when the homologous chromosomes, or the sisters, come apart. So it's the pulling apart of the genetic material in meiosis 1 or meiosis 2. So if we think about what can go wrong in meiosis 1, normally is what we want to do is take each of this pair of homologous chromosomes and pull one in one direction and one in the other. And that's normally what happens, and that's called disjunction. The chromosomes disjunct, they separate from one another. So for some reason, at a very low frequencies, what we can see, let's get a pen to highlight this really quick. There we go. Is what we sometimes see is that two or a pair of homologous chromosomes will actually go in the same direction. So rather than going like this, away from one another, they could go in the same direction. And so what then happens is that when this cell divides, the cell on the left has got a chromosome too many, and the cell on the right has got a chromosome too few, and that can lead to genetic defects. And so if we follow this all the way through meiosis 2, we're going to see this sequence of events in meiosis 2, this sequence of events in the other cell produced by meiosis 1, and so then is what we see is that some of the cells, like these two cells here, are going to have one too few chromosomes, and then these ones on the left are going to have one too many chromosomes, and so they're said to be n plus 1. N is the normal number of haploid chromosomes, or the haploid number of chromosomes. So you can be n plus 1 and n minus 1. So if you're n plus 1 and you get fertilized by a normal sperm, if you've got an egg that's n plus 1, you get fertilized by a normal sperm, rather than having 2 copies of each chromosome, you get one from mum and one from dad, if the egg has got 2 copies, then you can end up with a situation like trisomy, where you have 3 copies of the chromosome, and that can lead to genetic defects. So that wraps us up. That's it for mitosis and meiosis. I'll see you next time when we start talking about Mendelian genetics.